Hopefully everyone signed in okay. My name's Hill. I'm with SuperNOVA, and if this is your first time joining us for our program, we do lots of science outreach for youth across Atlantic Canada. Today we're focusing on ocean science, and it's actually Oceans Week, Canada and Halifax. Yesterday was World Oceans Day, so I think it's a good time to celebrate some of the cool things that we have in the ocean. What we're going to do today is a virtual fish dissection. And in your registration link, you should have been sent out an outline of a perch. That's just like a drawing. And what we're going to do today is, as I dissect our fish and show you the different parts of it, I'm going to get you to fill in your fish outline with the drawings of the body parts. So if you haven't already downloaded that, I'll just give you one moment to find it. And if you don't have an outline, you can also just start by drawing a kind of blob outline for our fish. Okay, so I'll be prompting you on what to draw as we go through it. But if you haven't already got a piece of paper and a pencil, I would grab one now. Now, I saw some folks talking on the chat. That's great. We want you to ask us lots of questions and let us know if there's anything that you couldn't figure out. But we're going to make sure that we keep everything on subject on the chat, so please don't use it to talk with a friend who you haven't talked to in a couple of weeks. You can do that after. But keep it on task, I guess, or on topic with oceans, okay? And if we do have people that aren't respecting that rule, we'll have to ask you to sign off of the webinar. We also have Laurel on as a moderator. Her logo is the SuperNOVA symbol, and she'll be able to message and answer questions if I'm not able to get to them, and she'll also be making sure that people are respecting that rule, that we're staying on topic, our chat. So I guess we can get started with our dissection now. And we actually use dissections to study animals for a lot of different reasons. Maybe if you have an idea of why we might dissect animals, you can throw it in the chat. But one of the main things that we can learn about different animals and about ourselves is by studying anatomy, and that's studying the body parts of different organisms. So often when you see an animal in the wild, you can see some of its external features, which we'll go through in a while, but you can't really see anything that's on the inside that really makes it live, like its lungs and its digestive system and all of those other things. So sometimes we do have to just dissect animals to understand them a little bit better. But of course, we don't want to be dissecting tons of wild animals that are living out in nature. We want to do it with as little impact as possible. So the fish that we have today is a perch. And this is a really abundant species, which means there's tons of them. And they're even a little bit invasive. And that means that they can go into habitats that they don't usually live, and they can actually succeed quite well in there. And that's fine, but sometimes perch are so good at succeeding in new habitats that they can actually kind of outlive and outcompete animals that naturally live there. So we call them an invasive species, and that's why this is one species that's kind of good for dissecting, because we know that we're not going to reduce the population by taking them from the wild for dissection. And in fact, if we take them from areas that they don't typically live, we may be able to help the ecosystem a little bit. It's important that even though we are going to cut into this animal and investigate it, we want to be respectful. This animal was once living, as all animals on this planet are, and we want to be able to respect that life. So as much as it may be interesting to dissect your own animals, I think it's really good to do it in a lab setting, because there's some safety concerns that you want to make, and you also want to make sure that it's done with animals that are meant to be dissected, and so that doesn't mean taking anything from the wild. So what we're going to do today is we're going to start with the external anatomy, so I'll show you some of this, and then we're going to kind of cut a window into our fish in a square. So if you are going to draw along for this dissection, there's a few things. You're not going to want to draw this fin here, because we're going to kind of draw a window in, and we're not going to draw this plate here. So maybe hold off on drawing anything until we identify specific body parts, and then I'll kind of give you hints on what to draw. Now, before I dive into anything, I have gloves on, one of the most important safety gear that you wear in a dissection, mostly because the fish is preserved with chemicals that I don't want to get on my own skin. I'm also going to put on some goggles just to protect my eyes from the same chemicals. And I've got a lab coat on, so we should be set. And then some of the tools that I'm using today, I've got some dissection scissors, I've got some forceps, but we also know those as tweezers. And then I've got a little scalpel here and a dissection pan and some pins. So we should be ready to go. Does anyone have any questions before we dive in here? I think we're maybe good. Okay, so this is the time to start with your perch outline. One of the reasons why we're going to draw along with this dissection is because biologists in particular, they use drawings to record things that they learn. And one thing that we do a lot of is do lab drawings or kind of recreations of what we see so that we have a record of the different organs that we're going to see inside this animal's day. Because of course, once we do the dissection, we can't keep this. It won't preserve forever. So a lab drawing with labels is a good way to kind of keep track of what we've learned. So one of the first things about this fish that we're going to notice is that its shape. It's kind of teardrop shaped like this. It's got a pointy nose and it's got a tapered tail, which means tapered means it gets narrower at the end. And of course, the fins at the end are a little bit wider, but mostly the body, it starts wide in the middle. It goes narrower at the end and narrower at the head. So we call this shape fusiform. And this shape actually allows water to kind of slide over its body with the least amount of friction possible. So when we're talking about friction with water or air or a movement through a fluid, I guess, we call it drag. So if this fish was shaped as a big box, the water would kind of just push on its head and it would be hard for the water to travel around the corners. And so the fish wouldn't be very fast. But with its tapered head and its tapered tail, it's quite quick. So this is called a fusiform shape. You'll also notice that if I point kind of frontwards, the body of the fish is kind of squished inwards. It's quite flat. We call this laterally compressed. And again, this helps the water flow over it to reduce drag so that it doesn't take that much energy for the fish to actually travel through the water. Now, one of the next things that I'm going to show you is it's got a dorsal fin here. And what you may be able to see at the very top are some bones. Now, the perch is a ray-finned bony fish. And we call it a ray fin because these bones kind of come out in rays. So this can be one of the first things that you draw along with. These bony rays are distinctive of this group of fish. It's a little bit more evolved than other fishes without bones, for example, sharks. So they have a few different features, but we kind of suggest that this type of fish is a little more closely related to humans than say maybe a lobe fin fish or a cartilaginous fish. So sharks themselves, they don't have bones at all. They have cartilage, and you can actually just squish your own ear. Our ears are made out of cartilage, so that's what a shark's kind of bones would feel like. But this perch has really sharp bones. So draw some of those rays in your dorsal fin. And then you'll also notice they have a few pelvic fins here down at the base. These also have bones. So as I stretch them out, you can kind of see the shape and you can draw that along. And then they also have a pectoral fin here. And this one's a little bit more, the bones aren't as hard, but the rays kind of spread out a lot more. Now, these pelvic fins down here and the pectoral fins here, these are what allow the fish to steer. One thing about a fusiform shape or that laterally compressed shape is if there's no drag and say this fish had no fins, it would probably just spin in the water like this quite a bit. So the dorsal fin usually sticks up like this, and it's kind of like a keel on a boat. It keeps the fish upright so that it doesn't spin in the water like this. And then last but not least, we've got the tail fin, or what we call the caudal fin, and there's quite a bit of muscle here. And so this is the strongest fin, and it's actually used to propel the fish. So this is what makes it move fast. And then these fins down here are for steering, and this fin here is for balance. So if you haven't already started, draw the rays into your fins, and remember that they're bony fish. And I'm just going to switch my notes here so I don't miss anything. Okay, so the next part that we might be familiar with. I think it's all safe to say that we know this in eye. And This is an interesting eye because it's much more developed than a lot of the eyes that other animals have in the ocean. Animals like crabs, snails, sea stars, they may be able to detect light, but they don't have a fully formed eye like humans do that can actually generate pictures. But a fish's eye is actually quite evolved, and so they can see more similar the way that humans see, and they actually have this little eyelid here. So you can draw an eye on your fish, and you can draw the little eyelid too if you'd like. And then of course we've got the mouth, and I'm just going to see if I can open it with my finger a little bit. So this fish has jaws, and it has tiny little feet, or not feet, teeth right around the edge that you probably won't be able to see. They're mostly razor fish. But one thing that I want to explain about this type of fish is that it does something called gaping. So it'll actually open its mouth and swim around in order to catch food. And the reason why is if you've ever tried to grab something in water, when you reach in with your hand, the current that you're creating by putting your hand in water actually pushes the object away. So it's really hard to just reach in and grab something underwater. But when the fish opens its mouth, it creates kind of a pressure difference. So all kinds of water is rushing into its mouth when it opens it. And sometimes that'll pull the prey into its mouth. So it's called gape feeding. It actually is almost like a suction cup. So you can draw a little mouth on your fish if it isn't even there, if it isn't already there. And you could draw a little pointy razor kind of sharp teeth down there if you'd like. All right, the next thing I'm going to show you here, I'm going to need my forceps. There's a little flap here right before the fin, and it's really hard, but maybe you can see me pulling it up like this. And this is called the operculum. And when the fish is alive, you might actually see this moving up and down like this. And what it's doing is the operculum covers the gills. And so when the operculum opens, it's doing the same thing that the gaping is. It's bringing water into the gills, and then the water will rush over the gills so that it can absorb the oxygen and then transfer it to its blood. So this is our hard bony piece. It also protects the gills because the gills are a really important structure. But this operculum will be moving like this kind of throughout life. Now, you can draw the operculum, but when I start to dissect, I'm actually going to draw a line kind of up through here. So it might be best to leave it for now, and this will be part of the window that we're actually going to look in for the fish. Okay, I think that we're ready to start cutting. Actually, there's one more thing that I want to show you that's actually really cool. So what you might be able to see along the fish is kind of a dark line along their body, just above kind of the midline. I think it's a little bit better to see over on this side. So you can see this dark line running around, and we're not going to be able to see it. We actually need a microscope, but along this line, there's all kinds of tiny little pores or holes into the fish's body that have special sensing cells on them. And so this fish can actually detect pressure differences in the water or vibrations caused by sound, and allows them to kind of pick up signals from their environment that are similar to the way we hear sound and use it to kind of decide where they go or what kind of prey they're looking for, whether they hide. So this is actually kind of like a sensory structure along here. And I'm realizing now that I forgot, but if we look really closely on the top, the fish does have two nostrils here. And they do work kind of similar to humans' nostrils, but rather than scents or chemicals being mixed in the air that we detect, this fish would actually kind of detect chemicals through water from here, and these connect to its brain. Okay, so I think we're ready to start getting into the internal anatomy. If you haven't already, make sure you're putting the rays on your fins, your eye, and your mouth here. And we're going to start cutting. Now, maybe while I'm starting to dissect, because it's going to take me a minute to get it open here, we can brainstorm and you can throw them in the chat, different types of organs that we have in our bodies that we actually need in order to survive. So as I do that, I'm going to cut down the base of our fish here through some of the bones. I'm actually going to start at the other end. It's a little easier. And I'm going to start right here at the urogenital opening. And this is kind of a multi-purpose exit for the body. We'll talk about in a little bit. Now I'm cutting quite shallow because I just want to get through the skin, the scales, the bones, and some muscle. I don't want to cut into the organs quite yet. So I think we've got that. Did anyone put any guesses in for organ systems that we have?
Yeah, Evan is saying lungs and heart. Lila says stomach. Maddie says digestive system.
Nice. Okay, those are all really good ones. And they're all ones that we have, but there are some modifications of a fish that will do kind of the same thing. So just gonna get through here.
Brain.
Yep, they do have a brain. We're probably not going to see the brain today. It's quite small in a fish, and it's actually pretty hard to find in there. So now I'm going to kind of lift this flap a little bit. And I'm going to try and gently lift it without pulling too much. This might not be a good camera angle, but just give me 1 second. Okay, so we now can start to see the internal anatomy of the fish. So one of the first things that we'll notice, I kind of cut into it here a little bit, is this fish to me looks like a male or a not mature female. Now we're talking about the biological sex here, and we can usually tell sex by maybe the different body structures that an organism has, but there's also a lot of different things that determine sex, and that can include hormones and body parts and all kinds of different things. So what I'm looking at right here is part of the male reproductive organs. This is the testes. So I'm actually going to remove this because we need to be able to see some of the stuff underneath. And in a female, they instead have ovaries. It would probably be about 3 times this size. And if we were to open them up, we'd be able to see tiny little eggs. So we're just going to put the testes here, and we can kind of see some of the other parts here. I'm actually just going to get a tiny bit of water to rinse it out. So in females, the ovaries are quite a bit larger, and so they'll actually press all of the fish's other organs really down far, and they're kind of like squished into the bottom. So in terms of learning about anatomy, a male fish is a little bit easier for us to see what we need to see. Now, I'm just going to lift up these other body parts for a moment, because I want to talk about this big white organ here. Does anyone have any guesses on what this could be? And I'll give you a big hint. Humans do not have one. I'm also going to, while you're thinking about what it might be, I'm going to actually make a little tiny incision so we can see what's inside. And this would be a good thing to draw onto your diagram if you wanted to draw along with that. It's quite hard, but you can see that there's really nothing in it. It's actually kind of an air-filled sac. And so this is a unique organ that even sharks don't have. So ray fin fish or bony fish are one of the first kind of developments or animals to develop a kind of a sac like this, and this is called a swim bladder. So what happens is that this swim bladder can fill with gases or air, which we know is lighter than water. And so if a fish wants to go higher up in the water column, it'll fill the sac with air so that it floats better. But if it wanted to go deeper, then it might fill that sac with liquid or just empty it entirely so that the fish would be a little heavier and it could sink. Now, some fish have the ability to fill that up, gulp in air and fill it up, but this fish actually doesn't. Though the gas actually comes from the fish's blood. And so it might not be really good at moving from a really deep place to a really shallow place quickly, but over a slower period of time, the fish could kind of adjust the blood that's going to this swim bladder to determine whether it's going to float more or less. So this is the swim bladder here in the back, and you can kind of draw a rough outline on that. And we are going to draw a few things over it if you want to follow along, but this is one of the largest organs that we're going to see. Okay, just going to do a quick peek in here, make sure I'm showing you the right things. So, yeah, we've got a few things going on here. So first off, this lobe that we have is something that humans have, and it's probably one of our most important organs. It does so many different things. This is the liver, and it looks a little bit like a lobe, and it's funny, this liver actually looks quite similar to what humans have. We have multiple lobes on our liver, so you might see like two sections like this, but this is the fish's liver. It looks like it only has one lobe, and it helps filter the blood. It helps produce enzymes that break down food. It reduces toxins from the body, so it's working overtime kind of all the time to make sure that the fish is getting the nutrients it needs and getting rid of the things that it doesn't need. So you can draw your kind of liver, maybe overlapping your swim bladder a little bit up like this. And I would recommend labeling it, because if we're not writing the name of the different organs down, we might lose track of what we're drawing really quickly.
So Evan asks, what do we even draw? So all the things that Hill is telling you to draw at this point, otherwise it's going to get too cluttered. It doesn't have to be perfect, just kind of draw the general area of where everything is.
Exactly, yeah. So you could even draw an exact square on your fish, exactly where I cut, and then honestly, everything is going to look a bit like a blob. So you can draw kind of a big long blob along the middle for your swim bladder, and then you can draw kind of a smaller blob for your liver. And if you have colors, maybe you can even use colors to kind of distinguish. So this liver is a little bit dark colored or yellow, and then the swim bladder is almost entirely white. So if we kind of go underneath here, we can see the start of the digestive system, which actually, I guess the liver kind of is the start as well, but we've got the stomach in here. And the stomach is usually, it might have, on different fish, it might have a few lobes like this. They have a different stomach than humans. Our stomach is one big sac. It's about the size of a softball, but it can expand to be about the size of a grocery bag. If you ate a really big meal, I wouldn't suggest it. So this in theory could probably expand quite a bit more if the fish had some food. But sometimes they'll have a few other lobes. Now, the stomach is mostly for storing food, and the liver will actually put some enzymes and digestive juices into the stomach to help start digestion. So we've got this here, and then if we go even further, I'm going to have to make a few more cuts here, actually. Okay, so you can kind of see this darkish tube along the bottom, maybe. Sorry, my head's probably in the way. Oh, here's, yeah. And so this is actually, this is a node to their stomach here, actually. And then this end, there's a tube over here that kind of travels from the rest of the digestive system. It looks like a kind of long tissue, and this is their intestine. Now, humans have stomachs and intestines. Stomachs, like I said before, are for storing and kind of starting the mechanical digestion, but the intestine is actually where a lot of the nutrient absorption will happen. So for humans, when we eat food, it takes about 8 hours for it to travel from our mouth into our stomach and then travel all the way through our intestine. Our intestines in a human about my size, your intestines, if spread out, could fill an entire badminton court. It has a really, really large surface area. This fish doesn't have as quite a big of an intestine, but it is still quite a large surface area for its body size. And the reason why we want more surface area is because it gives it more area to absorb the nutrients that are actually in there. Okay. So just as a quick review, I think this is another kind of stomach sac, but it isn't filled. So we've got our swim bladder along the back, this big white organ. We've got our liver, kind of the lobe up front here. We've got a stomach underneath with a few different blobs. And these are called the pyloric ceca, which really means stomach cavity. So these are just kind of like little stomach compartments. And then we've got our intestine running along the bottom. And then don't forget, we had our testes kind of laying on top here. But again, those might be hard to draw because we've removed them. And if you want to draw the intestine behind there, you wouldn't be able to see them. Okay. So we just went through the digestive system. So it starts with the liver making enzymes. And that enzyme is put into the stomach where there's food, and then that food travels through the intestine where the nutrients are absorbed. And then once the food is just waste, it'll exit through the urogenital kind of opening here, a multi-purpose exit for the fish. Now, there's one other structure that I want to show you that's really cool before we make any more cuts. And that's the gills. So the gills are this gray structure here. And if you get really close, you might be able to see that there's kind of different separations in them. Are you able to see that? Yeah. And then there's also actually different layers. So I could take all of them out. I'm not going to take them all out, but there's actually 4 layers like this on each side of the fish. And you can see all these kind of feathery projections. And if you think about it, because they have all these feathery projections and 4 layers of them, that means that the gills also have a really big surface area. It means there's lots of area around them for water to touch. And everywhere that water can touch, the gills can actually absorb oxygen from the water. And so the bigger surface area you have, the more efficient you can be at breathing, or I guess extracting oxygen from the water. So we've got 4 kind of gill layers here, and I cut them out a little bit, but there's a big, like this operculum has kind of like a big long bone along here to protect them all. So you can draw your gills right up. It's quite close to the eye, you'll see, but right at the edge of kind of our window that we've cut. And we'll take a note that they're really fan-shaped and they're quite kind of feathery. So you might want to draw that into your drawing a little bit.
BR asks, what is the waste hole called?
The urogenital opening. I think sometimes it's called the cloaca as well. And there's other animals that have that, something like birds, they'll lay eggs and expel waste through the same opening. So even though fish and birds aren't closely related, they do have that similarity. Okay, are we good on questions for now?
I think so, yes.
Okay, I'm just gonna get my instructions for the next part. So what we're going to do now is we're going to do our best to find the heart. And the heart is actually located right behind the gills. So the gills are what take oxygen from the water, but that oxygen has to travel throughout the fish's body. And the way that they do that is with the circulatory system. And that's kind of similar to humans. We breathe air in, it goes to our lungs, and then our lungs have all kinds of blood vessels around them that absorb the oxygen, put it into our blood, and then ship it all around our bodies. So the same thing is happening. The gills are absorbing oxygen. There's all kinds of arteries that you can't really see, but some, maybe there's some dyed really at the base there. But I'm just going to remove all of these and see if we can find the heart, because the heart is actually located right behind the gills. One reason is probably because then the oxygen has to travel less area in order to get to the heart, where it can then be pumped around the body.
Claudia asked, when the fish eats, where does all the water it drinks go?
That's a good question. You know what? I don't know. So we might have to look that up after. I've also wondered that. When it comes to whales, they'll like actually kind of spit the water out because they have baleen. But fish like this one probably actually ingest quite a bit of water. But maybe we'll have to look that up after. So
That was a good question.
that was a good question. Okay, just bear with me for a moment while I try and get in here. The heart is a little bit harder because it's such an important organ that there's quite a lot of bone and muscle around it. So it's actually kind of hard for us to get in and see. I'm just going to show you this gill up close because you can actually see there's like a hard part here. So these are called the gill breakers. It provides structural support, but it also helps protect blood vessels that are going to absorb that oxygen. It's also known as a gill arch. So like one gill would be a gill arch. Okay, I think I see our heart. I'm going to get you to zoom in on it before I try and extract it, just in case I have a problem. So our heart is right in here. This organ right kind of nestled into the gills. And this is all bone around here, quite hard, and quite hard around here. And then it's quite tough. So the flesh or the muscle around it is really strong. And one of the reasons why is because our heart is a muscle, and so is the fish's. Can anyone think why our heart needs to be a muscle? You can throw it in the chat, maybe. Any guesses?
Not yet.
Not yet. Okay.
Oh, Olivia says so it can pump.
Exactly. Heart's most important job is to pump blood around, and by contracting and then releasing, it actually creates kind of a pressure difference so that the blood can be shipped through all the blood vessels. So hopefully we can actually extract this heart because it has two chambers, which is a development for organisms like this. Animals that we consider maybe like less evolved or maybe less developed would have a single chamber heart, but this fish has two. Just bear with me one moment. I don't think we're going to get it out. But maybe what you'll be able to see is right here. There's kind of a lobe. Can you see that? Right by my finger. The heart's right in here. And this darker lobe, I would believe, hold on a second, I'm going to have to double check on this. So it has two lobes. It has an atrium where deoxygenated blood goes in. And then once the blood's in there, it's getting oxygenated. And then it leaves through the ventricle, which is this lobe over here. And this ventricle is really, really muscular. Like it doesn't really give much when I press on it. And the reason why is because it's really muscular, and that's because the oxygenated blood leaves the ventricle and goes to the rest of the body. So this is where the big squeeze is happening that pushes that blood and pumps it around. So we've got the ventricle right here. All right, so I'm going to lay this back down so maybe you can get oriented again where we are. So our gills, they were here, and then we removed them. And underneath, we've got our heart nestled right in here. Now, our heart doesn't look like a heart shape that you would see. It looks kind of more like a circle. But if you were going to draw it, you could kind of draw a lobe like this and then a smaller lobe like this coming off. And that would indicate that it's got those two different chambers, one for oxygenating the blood and then one for pushing the oxygen-rich blood through to the rest of the body. Okay, I'm going to take a moment here for any questions, if anyone's got some. And I'm just going to switch our fish for a second to see if I can get the heart out of this one. So this is a fish that I had a practice on earlier. And we're going to go through and just remember the different pieces. See, I didn't take the operculum off this one, so we might be able to get the heart here. So again, maybe we remember what this big gas-filled sac is called. Humans don't have them because we don't need to float. Well, not usually. This is called a swim bladder, so it fills with air. We've got this big lobe, the most important organs. It's the liver. So it detoxifies the blood, it filters the blood, it helps produce chemicals that help digestion. And then in here we've got some pyloric ceca, or stomach chambers. We've got the testes down here, and then we've got the intestines way at the bottom. And this one, actually, the intestines are a little bit better intact, so you can see. And we actually couldn't see this on the previous one. Right here is your spleen. It sounds like a silly name for an organ, but we have spleens as well. And spleens also have a role to play in filtering blood to remove anything that's maybe toxic. And they also are really important in the immune system. So the spleen kind of produces the, maybe not antibodies, but blood cells and whatnot that you can use to fight off infection. So we've got those. I'm going to quickly cut into the operculum to see if we can get the heart out of this one.
Claudia asked, if the heart is so small, how can the heart pump the blood through the whole body?
That is a really good question. It's a really strong muscle, and believe it or not, the perch actually has one of the bigger hearts of any other, well, not any other organism in the ocean, but there's actually a lot of animals in the ocean that don't even have a heart. For example, sea stars, jellyfish, they don't really have a heart. They have kind of these, they're called coelomic cavities, but really it's just kind of like open space in their body, and they don't have blood, but they have kind of like a clear liquidy substance similar to water that they just kind of push around their body without any blood vessels, and that gets the oxygen to them. So one way I've heard of it described is animals that don't have a heart, it's kind of like a pop bottle, or just like an empty water bottle. If you were to shake the water around in it, that's kind of how water's traveling around other organisms. So when we get to animals with backbones, we call them vertebrates, they have hearts. And they, even though it might look small, they're very powerful. Our heart in our body is only about the size of a fist. So you can think, in the grand scheme of our body, that's not that big. Okay, so we're going to remove these gills again. Maybe with some scissors. We're going to see.
Maddie asked, do any animals without vertebrae have hearts?
I'm gonna have to think about that one. Yeah, they do. Like earthworms, they have a heart. They actually have several hearts. So earthworms are kind of weird because they have segments, which means they're divided into different sections. But in each one of those segments, they'll have multiple organs. So worms actually have many hearts and many kidneys for filtering their blood.
I think it's like 5 or 6 hearts worms have.
Cool. And you can kind of think worms are quite long, but they're not super strong. So they might actually need lots of hearts to help pump the blood all the way from one end to the other. I think that lobsters they don't necessarily have a heart similar to us, but they have some kind of system with closed blood, which other animals don't necessarily have. So they don't necessarily have like a fully formed heart, but they kind of have blood vessels in like certain chambers specifically for their blood like fluid. All right, folks, this might not work out for us here with the heart. It's a little, they're so well protected in here, it's actually quite hard to remove it intact. Oh, that's cool. So something I just extracted by accident here. Maybe if I hold it over the dark thing is easier to see. If anyone can guess what this is, I'll give them a high five across the screen. But this is a hard bone. It was not connected to anything else. And you might not even think that fish necessarily have this organ, but this is an ear bone. Did someone guess?
Claudia's got ear, yeah.
Nice, good work. So this is in fact an ear bone, and I don't think you folks are going to be able to see it because they're really, really tiny, but there's actually like little bumps all the way around the ear bone, and these bumps, the ear bone continues to grow throughout the fish's life. So each bump is another year in its life. So scientists can actually look at this ear bone, count the bumps on it, and know how old the fish is. And that's really important to biologists because if you understand what the average age of a population is, you can kind of predict how that population will do in the future. And another cool thing that we can do with an ear bone is that these bones, since they've been growing throughout life, including very early in the fish's life, they're made predominantly out of calcium like our bones, but they also have different minerals in them. And depending on where a fish grows up, they might incorporate different minerals in its bones as opposed to another place where they grew up. So scientists can grind these up and do a chemical analysis on them and actually find out where fish were born. Not necessarily all species, but with some they have this procedure figured out. And so they can find, for fish like sturgeon that spend some of their life in freshwater and some of their life in the ocean, they can actually figure out which river they were born in by grinding up their ear bones. So that was a little sidetrack from the heart. I think we have the heart here, but I don't think I did a very good job at extracting it in one piece. This is actually not the heart. I think we've left it behind. So maybe we'll have to skip that one for today. I think our best image of it was probably on this one. We'll just take one more look. And it's that organ right here, kind of nestled in amongst the gills. Okay, folks. I think that those are all the organs, not all the organs that the fish has, but all the ones that I know of, or that I have the skills to dissect. Hopefully you learned a little bit from that dissection, but I'll stay on here for maybe, I don't know, 5 minutes. If people have some questions that they want to ask, I'd be happy to answer them. I'm just gonna wipe my hands off first.
Luke asks, can you do a frog?
Can I do a frog? That's a good question. I don't have any frogs that are preserved. Sometimes that they do dissect frogs, and typically they use bullfrogs because, again, we don't want to just be taking frogs from the wild and using them for dissection. So science companies or kind of labs will use a specific type of frog that is invasive. And if you remember from earlier on, invasive species can kind of move to a new habitat and do really well, and sometimes outcompete the animals that originally lived there. So bullfrog's a good one of those, and that's usually the type of frog that's used in frog dissections. But we're not going to do one today. Yeah.
Evan asked, is the dorsal fin the fin on the top?
Yes, that is the dorsal fin. Yeah, we can go through the fins again. The pectoral fin is the one right here on the side, and then the pelvic fins are down here near the base. And we have a pelvis too. It's kind of like our area in between our hips. And then we've got the caudal fin or the tail fin in the back.
I think that might be it.
Awesome. Well, hopefully some of you folks followed around with your dissection drawing. If you want to post it on Facebook or Instagram, you can tag SuperNOVA and we'll be able to see it. So you can check out our Facebook or Instagram page, and we'll keep an eye on it if anyone wants to share their drawings with us. But until then, I guess hopefully we'll see you guys next week for our next Oceans webinar. And remember that it's Oceans Week this week, so check out, if you go to oceansweek.ca, that gives you a list of all of the online events that are happening here in Halifax for Oceans Week until June 14th, and there's lots of free ones that you can join in on. We did an ROV launch yesterday that you can watch the feed from. We launched it in the Halifax Harbor and recorded video under there. So I recommend checking some of that stuff out if you're really into ocean stuff. And if not, until then, I'll see you folks next time.